Right, good day Junior Tuckies, and welcome to this video that is going to be dealing with meiosis in the formation of gametes. My name is Mr. P. I'm going to be taking you through this section and hopefully we're going to have a much better understanding within meiosis which will help us prepare for our final exams. Okay, so firstly, if we look at the process of meiosis holistically, so we're looking at the whole process and getting an overview, we see that the whole point here is for meiosis to reduce the number of chromosomes in the cell, and we call this reduction division. And what this does is it counters the doubling effect of fertilization at the end of the day. So that's where meiosis is so important. Okay, it's where in meiosis 1 we have our diploid nucleus. So in other words, we've got our two sets of chromosomes, 23 chromosomes from the maternal contributing parent, and then 23 from the paternal, and then our 46 in total that we have in the nucleus. This needs to be reduced to 23 chromosomes, and that's obviously the role of meiosis. So we can see here that we go from a 2N nucleus to an N nucleus, and then, funny enough, in meiosis 2, it remains N number of chromosomes. So in other words, my 23 chromosomes remain 23 when I have the formation of the gametes at the end of meiosis 2. We'll see that when we count the number of chromosomes in a nucleus, we always look at the centromeres. And if we count the centromeres, we'll never count the number of chromosomes incorrectly. So keep your eyes on the number of centromeres, because we're going to be using this skill a little bit later on when we look at the worksheet questions. Okay, we'll see at the end of this meiotic division we find ourselves with those 4 unique gametes, and I use the word unique on purpose because there are a couple of things or a couple of processes that ensure genetic diversity in these gametes. Okay, so if we look at the gametogenesis, so this is the formation of the gametes through meiosis. In males and females, the process happens a little bit differently. In males, the formation of sperm we refer to as spermatogenesis, and in females, the formation of the ova we refer to oogenesis. And I've put the picture in there just to show you the difference in size, and you can for yourself now think that these two processes where we form the sperms, which you can see over there, and where we form the ova, which you can see over there, have to be quite different to produce such different gametes at the end of the day. All right, but we're going to be looking at these two processes in a little bit more detail in the next theory video, so make sure that you get your hands on that one. For the time being, we're going to be looking at the phases of meiosis, particularly in the general context over here. So if we look at prophase 1, and remember that the number 1 in this case is so important because it's the first meiotic division, and the phase here is prophase. So we're looking at the disappearing nuclear membrane, our centrioles that are moving to opposite poles of the cell, and that is important. I've underlined that on purpose there, so we can't just. So over there you'll see opposite poles is so important because the centrioles are moving in that direction. Chromosomes arranging in their homologous pairs, that's important. They don't just arrange in random pairs, they're in their homologous pairs, and then crossing over of homologous chromosomes. And it's not of chromosomes, it's of homologous chromosomes, because they are going to be in their homologous pairs. So crossing over of homologous chromosomes is what also ensures this genetic diversity at the end of the day. So that's the first one of the processes that we are going to identify later on when it comes to ensuring genetic diversity. Okay, so let's look at this image analysis quickly, and you can see that point number 1, the disappearing nuclear membrane, can be seen in the image over there. We can see the centrioles moving to the opposite poles, and point number 4, we can see that crossing over has taken place because the evidence in this diagram shows that there is a different color to each of those chromatids, which shows that genetic information has moved from one of the homologous chromosomes to the other. If we look at metaphase 1, we're going to do an image analysis now as well. We can see that the centrioles reach the opposite poles, and now we've got the formation of the spindle fibers. All right, we can see from the centriole to the centromere of those homologous chromosomes, we're going to find that little spindle thread or that spindle fiber. You can see the alignment of the homologous chromosomes is quite important here too, because the centromeres we don't find on the equator, they're on either side of the equator. So that's important as well. We can see that the chromosomes, like I said before, in their bivalent pair lie on either side of this equator, and they do this randomly. So there is a random arrangement to this, which means our maternal homologous chromosome and paternal homologous chromosome could be on either side. There is no rule to which side they're going to lie, and this random arrangement also adds to the genetic diversity at the end of the day. So in other words, what is going to occur or end up in the gametes as genetic information. So that's our metaphase 1. If we look at analysis further here, you can see that they're there randomly on each side. And it is quite important to link these points in metaphase, prophase, anaphase, telophase to a diagram, because it's very likely you're going to be getting a diagram in your exam that you need to analyze and identify the specific phase and the occurrences in that specific diagram. If we look at anaphase, the identifying characteristic here is the actual moving of these chromosomes now to the opposite poles. So these bivalent chromosomes are being pulled by the contracting spindle fibers, and we're going to find each one of these bivalent chromosomes at opposite poles of the cell. You can see that is where the equator or the equatorial line was, and those chromosomes are now being pulled from the equator. If we look at telophase 1, we can see groups of chromosomes gathering at the poles and the reforming of the nuclear membrane, as well as the cytokinesis taking place, which is the invagination that you see there of the cells. And we're going to have n number of chromosomes because now we're counting centromeres, okay, in the forming of the 2 new cells. You can see that we still have chromatids joined by the centromere, and the centromere has not been split yet. If we look at prophase 2, we'll see that here, this is one of the cells that we've just formed after telophase 1. And we're going to be looking at this cell in a little bit more detail just to analyze and get a better idea of prophase 2. So we can see we have the disappearing membrane again of the nucleus. We've got centrioles moving again to opposite poles, and each of these chromosomes consists of 2 chromatids joined at a centromere. And what's going to happen now is that in meiosis 2, we're going to have the separation of the chromatids at the centromere. So that doesn't happen in meiosis 1, it's going to happen in meiosis 2. Right, let's have a look here. Metaphase 2. So now we've got M for middle. So now we've got the arrangement of these chromosomes on the equatorial line. And this new equatorial line is going to be perpendicular to the previous one. So we can have meiosis 1 and 2 happen simultaneously in cellular division. Sometimes you'll see a diagram like that. In this case, I've opted to just use one cell, and you can see here that now the centromeres are actually on this equatorial line. Once again, the arrangement of these chromosomes could be either side of this equatorial line. So there's no specific chromatid that will form on either side. It is completely random. Our spindle fibers again will attach to now the centromere of these chromosomes, and on each side they are going to split. Now I've jumped ahead now to anaphase 2, because as these centromeres split, we enter this next phase, anaphase 2. Now the random arrangement of chromosomes takes place in metaphase 2. But I've put it here because now this random arrangement has led the spindle fibers pulling each of these chromatids randomly to opposite poles of the cell. And you can see that happening over there. This is going to mean that quite randomly, these chromatids are going to form part of the new haploid cells, these haploid gametes that are going to form, and the chromatid that now exists inside of these gametes is genetically unique because of the randomness in the process of meiosis. We can see there's one chromatid. There is no second chromatid joined at the centromere here. So now we actually refer to this unreplicated chromosome as exactly that, an unreplicated chromosome that is genetically unique. Well, Junior Tuckies, I hope that this quick run through of the process of meiosis has helped you better understand each of the phases in meiosis 1 and meiosis 2. I do hope that you tune in for more of the videos and to get a better understanding in preparation for your exams. Make sure you do get your hands on the theory booklet as well as the worksheet with a worksheet memorandum for some of those practice questions so that you're well prepared at the end of the day. I do hope to see you again. I hope you tune in. Thank you. Have a good day.